Hi, I'm Dan Knights. I'm going to be discussing integrating diet and microbiome for longitudinal analysis of the infant microbiome. Disclosures, I'm a scientific advisor at Differentiation, which is a company involved in commercializing microbiome analysis, although everything I'll talk about today is all open source and peer-reviewed. So first, we're going to discuss linking longitudinal diet data with longitudinal microbiome data. And then we'll go into some more general longitudinal analysis methods. And I want to start with this shotgun metagenomics data that was collected by Abby Johnson. She was a postdoc in my lab at the time and is now a tenure-track faculty at the University of Minnesota. She collected these daily shotgun metagenomics samples from 34 people over the course of 17 days. The study was sponsored by General Mills. And we have here visualized the species level variation in these people over time. So every color represents a different species. It's actually the first time that we were ever able to visualize this on a daily basis for a cohort of people, because we did such dense shotgun sequencing. And each of these boxes shows you that person's kind of profile and stability. So you can see some people are more stable than others. This is Abby's microbiome. She had the most stable microbiome. And some people are also quite different from each other. So like, you know, this subject has no microbiome that's similar to them. These three people on the right each have a high abundance of Prevotella. So you can see everyone kind of has their own fingerprint. Some people are a little more similar to others, and some people are a little more variable than others. Then at the same time, we collected dietary data. Now that's what I'm showing at the bottom. And in this case, it's colored at a very high level. So we have grain products, sugars and sweets, milk products, and yet you still see very high variation from day to day. In fact, the diet data is more variable than the microbiome data, and also highly individualized, especially when you zoom in on it. There were two subjects who had some special circumstances. They decided to consume only Soylent for the entire study. This is a meal replacement beverage. And even though they did this, and even though they were cohabitating, they did not converge. So their microbiome stayed distinct, as you can see here. And their microbiomes also didn't stabilize, which was kind of interesting. So, so what we're going to do first is to forget about all this day-to-day variation in each person and just try to find out if the average similarity in people's microbiomes, which is shown on the left, corresponds to the average similarity of their diet, or their dietary intake, shown on the right. Each person here on the left, each sample from each person is colored by individual. And you can see the samples from one individual cluster together, so everyone stays kind of in their own microbiome cloud and moves around in that space over time. And some people are more similar than others. So what we're going to be looking for is, if you measure the distance between the centers of two people's microbiome clouds, is that associated with the distance between the centers of their diet clouds? If it is, then that means that diet and microbiome are associated or corresponding here. But how do we analyze the diet data? We found actually that the diet data were higher dimensional and more sparse than the microbiome data. This again is Abby's microbiome. Abby's diet and microbiome. The diet's shown at a very high level. When we go down to specific foods, it looks like this. So now it's very challenging to analyze. Most days have almost many different foods from the previous day. We took a minute to zoom out and examine and visualize just the diet data. So there's no microbiome data here. And this is a network visualization that Abby made, where each of the white points is a person, and then the small colored points are the foods that they're eating. And the foods are arranged to be close to someone who's eating them, but also far away from someone who's not eating them. And what you can see is that there are some foods in the middle, like this is dairy, bread, that are eaten by most people or many people. But then there are other foods on the periphery, most of the foods, that are only being eaten by 1 or 2 or 3 people. You can see the Soylent drinkers themselves are on the periphery here. One of them had pizza one time during the study, which is why they're connected in with the main group. What this shows us is that the diet data are indeed highly personalized and highly sparse. We don't have a lot of foods that are consistently eaten by a lot of people. So what do? How do we analyze such a sparse matrix? Well, the most common approach to dealing with this is to instead convert the food profiles into nutrient profiles. And we have these for all of the foods in our database. There are about 100 nutrients that are tracked, and these are macronutrients like carbohydrates and micronutrients like all of the vitamins and minerals that we normally track. So, so we projected the foods down into nutrients, took the average nutrient profile, and then we're going to see if that corresponds to the average microbiome profile. On the left here, I'm showing the average microbiome distances between people. And on the right is the average nutrient distances between people throughout the study. So each point represents one person's average throughout the whole study. And we're going to do what's called a Procrustes analysis, where we rotate one to see if it lines up with the other. And if it does, that will mean that there's a correspondence. And unfortunately, when we tried this, we found they really did not line up. And we tried this in many different ways, different types of normalization for the nutrient data, different distance metrics for the nutrients for the microbiome. And we could not get them to correspond. And we thought about this, and we realized it actually kind of makes sense. The microbiome did not pair well with the conventional nutrients, but we know that the conventional nutrients are ignoring the vast majority of what's actually in the foods that we're eating. So for example, these are all of the chemical constituents of a strawberry, and there are all kinds of things in there that probably matter a lot to your microbiome. These are different types of fibers, different sugars, different fatty acids, different preservatives, different coloring agents. And yet they're completely ignored when we try to use standard nutrient profiles. Abby fortunately came up with a way of kind of cutting around this, because on the one hand, everyone's eating unique foods, so we can't use those. On the other hand, the nutrient data are missing all of this dark matter. She found a kind of a middle ground where she was able to use the hierarchical structure of the foods in our food database to build a tree of foods. And what this is doing is putting foods that have probably kind of similar dark matter to each other, near each other in the tree, so that we can share information between them. And that means, if we look in the tree and we want to compare, say, one person who's been eating spinach to another person who's been eating kale, we can consider their diets to be a little bit more similar to each other than they are to someone else who's been eating an egg, for example. Now we're going to go back to the same question. We still have the distances between everyone's average microbiomes. And here we're going to try to compare them to the distance between their average food intake, using the food tree to compare what they ate. So the question here is, are people who ate similar foods in the food tree distances going to have more similar microbiomes? And when we did the same analysis, the Procrustes analysis, we found that yes, in fact, they did line up. And this is the only way that we were able to find a correspondence in this data set between people's average dietary intake and their average nutrient intake. So we found that the foods, rather than the conventional nutrients, were better at explaining the microbiome variation. It was a Pearson correlation of 19.4%, which means that the foods explained about 3.8% of the microbiome variation between people. So now let's zoom in on each person, and we're going to take a look at the longitudinal aspect using a similar method. And the question here is, when someone's moving around from day to day in their microbiome cloud, does that correspond with how they're moving around in their diet cloud? And we're going to use exactly the same type of analysis, except here we're going to look at all time points for one subject. And that's on the left side is their microbiome. And we're going to try to overlay that on all of the time points for their diet. And again, if they correspond, it means that when they eat similar foods, their microbiome tends to have a similar profile. When we did this, we had to take into account the last few days of dietary history to get a significant result. And doing so showed us that that it was significant in the almost all of the subjects. And we're going to visualize this by looking at the first axis of this Procrustes analysis plotted over time. So you can see the diet and microbiome kind of moving around together. And these were significant for almost everyone in the study. And what we saw here is that diet explained about 9% of the microbiome variation within each person. This was a Pearson correlation on average of 0.24. So it seems as though diet is actually explaining about twice as much of the within-person variation longitudinally as it is of the cross-sectional variation between people. And this may be one reason why we haven't seen a lot of strong diet microbiome correlations in cross-sectional data. Because we had this longitudinal data, there's this other cool thing we were able to do, which is we can actually ask for each person whether specific foods are correlated with specific microbes over time. This only works if it's a food they ate frequently at differing amounts. And we saw that some of these associations were quite generalized. So for example, this cakes, cookies, bars group was positively associated with Roseburia inulinivorans in multiple people. And this kind of makes sense because inulin is a common fiber additive in a lot of cakes, cookies, and bars. So, so that might be a biologically plausible association. However, the vast majority of these associations, and we found many of them, were not conserved across people. So they would be negative in one person and positive in another. And this again shows why if you did a cross-sectional study to associate diet with microbiome, you would never find these associations. You could have 10,000 people in your study and you would never find some of these associations because they're going up in some people and down in others, and they'd cancel out. So another benefit of longitudinal data. Now, I'm going to shift gears a little bit and talk about a more general tool that was developed in my lab by Robin Shields Cutler, who was a postdoc in my lab at the time and is now tenure track faculty at Macalester. And this is a tool called SplinectomeR, and it's used for testing for differences between groups, and specifically in longitudinal data. So the idea is that you'll often have these shapes or trajectories of a microbe's abundance over time that are not going to be linear. If you look at a particular microbe's abundance in infant over time, it doesn't just follow a linear trajectory. It has some sort of arbitrary shape. And we wanted a way to be able to test whether that shape was different between different exposure groups in our data set. So this tool is called SplinectomeR because it fits a spline to the trajectory in each subject. And then it looks for differences, systematic differences between all of the splines in one group of subjects and all of the splines in another group of subjects. And it has 3 different ways to test this. You can ask whether the whole trajectory is different on average. You can also find a short window where there's a difference. And you can also ask whether a trajectory of one feature is like increasing over time or decreasing. So the first one is called Permuspliner. And we applied this to an infant microbiome data set, longitudinal analysis from Moran Yasur. And we reanalyzed this with SplinectomeR using the Permuspliner function. We were able to find that this one family, Porphyromonaceae, was strongly differentiated between the antibiotic exposed infants and the non-antibiotic exposed infants in the shape of its trajectory over time. So the average trajectory in the antibiotic exposed infants is shown with this solid curve on top, and then the average in the non-exposed group is shown at the bottom. You can see in the background all of the splines that were fitted to the permuted, to the data when you permute the class labels. And this is highly significant. You can also just visualize the distance between the groups over time. That's what's shown on the right. So that was a new finding on those data. We also looked at Bacteroides, which was found to be different in certain aspects according to antibiotic exposure in the original publication. So we ran Permuspliner, this function, to test whether the overall curve of Bacteroides was different after antibiotic exposure in the infants, and it was not. But we also ran this other function called sliding spliner that tests whether a window of time might contain a significant difference between the groups. And here we found actually that right around 30 months of age, there was a significant difference between the trajectories of Bacteroides in the 2 groups. And you can see here that the p-value of significance is dropping in that window. So this is kind of nice when you have a data set where you might have arbitrary time points, missing samples, and longitudinal data, and you don't really know the shape of the curves over time, and you don't know whether the significance is going to be, in the whole curve, or maybe just in a certain window of time. And then the final tool in this, or function in this, is called Trendyspliner, which just tells you whether a trajectory is increasing over time or decreasing over time. For example, we saw that alpha diversity trajectories were not different between antibiotic exposure groups, but alpha diversity is increasing over time in the infant group. And we know that from visualizing it, but this is a statistical way to test this, even when the shape is not necessarily linear. So it's kind of a neat tool for doing these types of analyses. Very flexible and powerful and quite simple. So I encourage you to check that out. And just to conclude, I discussed how we found in our diet microbiome study that foods, rather than conventional nutrients, explain most of the microbiome variation. We saw that dense longitudinal sampling can show that there are these personalized diet microbiome associations that would be masked if you just run a cross-sectional study. And then finally, I presented SplinectomeR, which is a nice tool for comparing arbitrary trajectories of microbes over time, and even finding windows of significant deviation between groups in your study. And I'd like to acknowledge everyone who contributed to this work, and thank you again for your time and attention.